Hi everybody, welcome to Vijayasof Foundation. My name is Manoj Swaminathan and today we are going to learn about the role of pharmacogenomics in pharmacovigilance. But then, before we get started, I had a question for all of you. What would happen if all of us and our family members received the COVID vaccine based on our medical history or genetic profile with guaranteed more than 90% efficacy and safety? Why am I telling you this? It's because in the market there are so many varieties of COVID vaccine available. Now, what if you get the vaccine which is suitable for your body? Something which will definitely work and give you the required efficacy and safety, right? So the example is this way, where a family, or rather I would say an extended family, with all the persons in the family getting a different vaccine based on their body structure, genetic profile, medical history, and the safety profile. Based on all this, they get the appropriate vaccine. If this would have happened, then it would have been so beneficial, right? And that brings us to today's topic, and that is about pharmacogenomics and patient safety. Now, I'll give you some background to this topic. There is a large variability in the response to drug therapy, both in terms of efficacy as well as safety. The same medicine, if given to two people, both of them will respond differently. One may have an adverse event, other may not have an adverse event. For one, it will be extremely effective. For the other, it may not be that effective. The same drug may induce an adverse reaction in some patients and not in others. Let me give you the example of COVID vaccine. I and my friend, both of us, took the same vaccine on the same day. The next day, I had fever and body pain. However, my friend did not have any adverse event, nothing. When we took the second dose, I did not have any adverse event. However, my friend had fever and body ache. Now, the same vaccine, also probably from the same batch because both of us took the vaccine at the same time, and both of them reacted differently. The genetic variations among patients that may relate to drug disposition or drug action, right? So the genetic variations will be there. So based on that, the response will be different, okay? And both in terms of pharmacokinetics as well as pharmacodynamics. So how do you define pharmacogenomics? It is the study of how genes affect a person's response to drugs. So we have pharmacology, which is the science of drugs. And then we also have genomics, which is the study of genes and their functions. And when you intersect, you get pharmacogenomics, right? And that is about safe and effective medications as per the patient's genetic makeup. So you will get the medicine which is suitable for you, or you will get the vaccine which is suitable for you. So it's all about prediction, right? And that's also about the safety profile, wherein you predict whether an adverse event will happen or not, right? So when you talk about the pharmaceutical industry, it is primarily product centric, okay, where a drug is manufactured and then you look at the potential sales of the medicine and also how much or how can you scale it up and which all countries you want to commercialize and so on. However, things are going to change in the future. With the advent of pharmacogenomics, we are going to move from being product centric to patient centric, and that is pharmacogenomics. Why patient centric? Is because you would have a tailored medicine for every patient, right? So, whatever is suitable for the patient, only that medicine they will get. So, what do you mean by personalized medicines? It is tailoring of medical treatment to the individual characteristics of each patient. Right? What does a tailor do? He or she will stitch the clothes as per your body structure. Right? Something which will fit on you. Something similar will also happen with medicines, wherein you will get medicines which will be suitable for you. Okay? Personalized medicines are also called as precision medicines. Please remember this. Personalized medicines are also called as precision medicines, and these form the basis for pharmacogenomics. Okay. Now, the what is the approach of personalized medicine? That it is based on the molecular or genetic profile of the patient, and as compared to the susceptibility, right? And based on this, you decide on the ideal treatment, and then you administer the tailored medicine to the patient. So, in this process, you make the medicine safe as well as effective. Why safe? Is because the likelihood of adverse events may go down, and effective is because based on the genetic profile, you know the medicine is definitely going to work. So it's all about the right treatment for the right patient. And that is pharmacogenomics. Right treatment for the right patient. Otherwise, what would happen? At least we saw during the COVID vaccine times, that same vaccine was given to the people who enrolled, got into, and like they were administered the same vaccine. Nothing based on the genetic profile or nothing. We didn't even think about whether the vaccine is going to be suitable for them or not, right? So in future, things may change. So I have a very interesting video for you, and this is about precision medicines. Okay, now let's get started, and I will also make it full screen so that it is useful for you. Okay, now let's get started.
Precision medicine. You've probably heard the words on the news or even at your doctor's office, but how does it affect your life right now? Precision medicine is basically a modern approach to understanding health. Take asthma. Asthma affects more than 25 million Americans, nearly 1 in 12. They all have one thing in common, a condition that makes breathing difficult. In most clinics, these patients are treated with an inhaler and the same drug, albuterol. But are 25 million people all the same? If you have asthma, your condition depends on where you grew up, how old you are, and your ancestry. It also depends on the air you breathe and if you have carpets or pets. Every patient is unique, so one-size-fits-all remedies don't always work. It's not just asthma. For diabetes, cancer, and most other diseases, today's treatments are based on obvious symptoms, standard tests, and common drugs. Precision medicine, however, starts with creating a clear picture of health by collecting, connecting, and analyzing many kinds of information. This could be data you provide from your fitness tracker, or it could be what scientists are learning in their labs right now about the underlying causes of disease. Genetics, your health history, and other details are added to the mix. When we integrate such data from millions of people, your clinician and researchers can understand your health risks, how others like you responded to a disease or drug, ways to prevent disease, and what makes you unique. So we can stop focusing on diseases and care for you as an individual. The more information we collect and connect, the better scientists will understand health. And as that knowledge network grows, we can ensure that every patient everywhere gets the right treatment in the right dose at the right time.
We will now learn about the pharmacogenomic biomarkers. So these are something which decide on whether the medicine will work in the patient or not. Again, for this, I have a very interesting video for it. And once we are done with the video, I will be able to explain to you in detail. So let's get started.
Biomarker is short for biological marker. It means any sign of biological process that can be objectively measured. Biomarkers indicate when and to what extent a physiological process is taking place and what is going wrong in the body of a sick person. Thus, they can be used both for diagnosis and for monitoring the success of a therapy. A familiar example of a biomarker is the detection of sugar in the urine of diabetic people. 2,500 years ago, two Hindu physicians were the first to describe the sweetness of diabetic urine and to use it for diagnosis. Ants are attracted to anything sweet, and the physicians pointed out that if ants were attracted to a person's urine, it meant that person had diabetes. Today we have simple and reliable tests which can detect and quantify sugar in blood. No need for ants anymore. In drug discovery and development, biomarkers of all types play a big role. It can be something as simple as blood sugar or as sophisticated as the presence of a certain genetic mutation in the DNA of a cancer cell. The use of biomarkers in clinical development delivers information on the efficacy and safety of a treatment, and ideally indicates which patients benefit most from a potential drug. Indeed, it is quite evident nowadays that not only does each person have a different genetic makeup, but also that each person's disease is different even when they have the same diagnosis. Biomarkers, single or in combination, can provide detailed information on the hidden identity of a disease and the best way to tackle it. This is the cornerstone of personalized healthcare, a vision of medicine where each patient receives a therapy tailored to his or her specific genetic makeup or disease subtype. At Roche pRED, biomarker research employing modern analytic and gene sequencing technologies is an integral part of the discovery of a new medicine. From the first step of target identification to the most advanced clinical development plan. A biomarker can be a specific tumor gene, a revelatory neurotoxic peptide that spills from the brain into other parts of the body, a unique viral protein in infected blood. In collaboration with Roche Diagnostics, relevant biomarkers may be developed further to become marketed diagnostic tools, on the way towards a perfect fit between therapy and patient.
Now, the biomarkers in various diseases, right? So what we learned about biomarkers is it is disease specific and also it is patient specific in terms of availability of those biomarkers in the patient or not. So this is an example, okay, where you can find the various biomarkers and also in relation to the drug and what can be the clinical response. Some examples include that of isoniazid, which is an anti-tuberculous medicine, and for that you need to evaluate whether the patient has got the NAT2 gene, and if the patient has got that gene, then it is very possible that the patient may develop isoniazid toxicity or even develop systemic lupus erythematosus (SLE). So you can prevent adverse events just on the basis of the genetic profile of the patient. Okay, so research is going on, and we will have more and more biomarkers in the future. Now let us learn about biomarkers with an example. Right? It's the best way to learn pharmacovigilance, and that is with the examples. And the example which I have for you today is herceptin or trastuzumab. Okay, so this medicine, which is used for breast cancer, I'll explain that in detail. Okay, and this is considered the showstopper for say genetic or pharmacogenomics, or I would even say personalized medicine. Okay, and now there are many more medicines coming into the market, and this was the first one to do that. Okay, that is trastuzumab or Herceptin. So before we proceed, you should remember the name of this biomarker, and that is HER2. Okay, that is HER2 biomarker. The full form is human epidermal growth factor receptor 2. Okay, HER2 biomarker. Okay. Now, what is Herceptin? Herceptin is a genetically engineered humanized monoclonal antibody that targets the HER2 biomarker. So, in the previous slide, what did I say? That I spoke to you about the HER2 biomarker, and if the patient has this HER2 biomarker, then it is quite obvious that the medicine will work in the patient. Okay. Now, what is Herceptin used for? Or what is trastuzumab used for? What are the indications? This medicine is approved for the treatment of early breast cancer, that is HER2 positive, that is the patient has HER2 receptor and has the spread into the lymph nodes, or is HER2 positive but the disease has not spread into the lymph nodes. In both these conditions, this medicine will be effective. Okay, so that is the basis. Now, again, I have an interesting video for you, and this is about the anti-HER2 uh, biomarker medicine. Okay, and this is all about trastuzumab. Okay, now let's get started.
Anti-HER2 mechanisms of approved HER2 inhibitors. The development of many breast cancers are driven by the presence of HER2. HER2 is a transmembrane tyrosine kinase receptor which belongs to the family of the EGFR, epidermal growth factor receptor. Its unique feature, which differentiates it from the other members of the family, is the absence of a known ligand. HER2 is overexpressed in 25 to 30% of human breast cancers and has been determined to be an adverse prognostic factor. Since the level of HER2 in human cancer cells with membrane overexpression is much higher than in normal adult tissue, there is potentially more sensitivity to the toxicity of HER2 targeting drugs. HER2 overexpression is typically found in both the primary tumor and in metastatic sites, which provides the rationale for the effectiveness of anti-HER2 therapy at all disease sites. Trastuzumab. Research has therefore focused heavily on HER2 inhibitors as anti-cancer agents. Trastuzumab is the first of such agents which was registered for use in patients with HER2 overexpressing breast cancer. Trastuzumab is a recombinant monoclonal antibody directed against the extracellular domain of the tyrosine kinase receptor HER2. Trastuzumab has shown clinical activity in HER2 overexpressing breast cancers, and at present is currently approved for patients whose tumors have this abnormality in both the metastatic and the adjuvant setting. A number of different mechanisms of action has been attributed to its anti-HER2 activity, including antibody-dependent cell-mediated cytotoxicity, prevention of HER2 truncated membrane-bound fragment following HER2 overexpression, and HER2 receptor downregulation, though these are still subject of discussion. However, binding of trastuzumab to the HER2 receptor and subsequent inhibition of HER2-mediated signaling transduction is thought to be the main mechanism of action of trastuzumab.
I hope you found that video to be useful and interesting. So it is like a God-made medicine only for the patient, right? And you know that it is definitely going to work on the patient. So what are the advantages of personalized medicine and even pharmacogenomics? First is you can decide on an optimized treatment, right? And that will be tailored for the patient, okay, as per their needs and as per their biomarkers. And this will also help in the reduction of the treatment duration, because you are not experimenting and you know that the medicine is going to work, so the treatment duration is going to be lower. Then the healthcare costs will gradually go down, and it's all about pharmacovigilance, right? So the incidence of adverse events is also going to go down, right? Because you know the patient is going to be benefited, and there may not be any toxicity as a result, and hence there may be a reduced incidence of adverse events. I don't say that there won't be any adverse events at all, but then the number of adverse events will definitely go down. In the end, this is going to help with the new drug approval time. What does that mean? That new drugs can come into the market quickly. So that is going to be the greatest advantage of pharmacogenomics or personalized medicine. Okay, so that's something we should always remember. Now, the best part is that there is improved benefit risk balance, right? Because ultimately you know that the chances of adverse events are going to go down, right? And you know that the patient has the required biomarker, and you also know that yes, the patient is going to be benefited. So that's why pharmacogenomics has a very good role in pharmacovigilance. However, there are some limitations when we talk about personalized medicine and also pharmacogenomics on the whole. There can be delay in biomarker testing. How about the developing countries which do not have the required laboratories for identifying the biomarkers? That can be a problem. What if in some countries there are only one or two labs? That can again be a problem because you need to transport the medicine, the sample to that lab and get it tested. So there can be delay. There can be lack of prospective clinical trials. That's again a limitation. There can be non-genetic factors. So something related to the, I would say, the patient behavior, the diet, environmental conditions, and so on. So those may also have an impact. At the same time, you may even consider concomitant medications or the co-suspect medications. They may even have an impact. Okay, so there can be some non-genetic factors also which can impact. At times, there can be insufficient predictability of the biomarker test for recommending the genotype guided dosing. So these are rare times, or this can happen. Okay, so again, this can be a limitation. So what is in store for the pharmaceutical companies? What do you think? Can they increase the prices? Can they decrease the prices? What is in store? So let's talk about both the positive aspects as well as the negative aspects. So let's start with the negative aspects, that the sales can get impacted, right? Now, if they are manufacturing one tablets in a year, now they know that the patients are less, so they may need to only manufacture 10,000 tablets per year. So from 1 million, the number of tablets go down to say 10,000, right? So that's a great impact on the sales. Then clinical trials, they need to spend on the clinical trials, and this might be slightly expensive, but then at the end of the day, they are going to get early approval. So again, they may need to consider this. Then the local infrastructure, right? I was talking about the labs. So even if the product is in the market, do we have the required labs which can identify the biomarkers, right? So you need to think about all those things. However, there can also be some positives wherein the company can have early approvals, right? So that's what we discussed in the previous slide, that there can be early approvals because of this. High prices. So what does this mean? So imagine the company sells one tablet for $1, okay? And if they are manufacturing 1 million tablets, then they are going to get $1 million. But now, because of pharmacogenomics, you know that only 10,000 patients have that biomarker, and hence you have reduced the manufacturing. Now, the company still wants to make money. So what they will do is they will charge around, say, $100 per tablet. So from, say, $1 per tablet, they will now charge you $100. So still they will make the same amount. That is $1 million. So that way the prices may go up. This may have an impact even on the insurance. See, at the end of the day, we are learning pharmacovigilance, right? And the number of adverse events are going to go down. So from pharmacovigilance point of view, it's a win-win situation. Now I'll talk more about the relevance to pharmacovigilance. First and foremost, we are talking about personalized medicine. So we know that the medicine is going to be effective in the patient, right? And the chances of adverse events are going to go down. Then, as I told you, the number of adverse events are going to go down. And you can also predict whether the patient would develop adverse event or not. And at times you have no option, you don't have other medicines, and you have to give this medicine, so you can directly target the adverse event. An example is a medicine, considering the patient's biomarkers, is going to cause liver toxicity. So you know it is going to cause liver toxicity. The moment you give the medicine, you start monitoring the liver function test and you take care of that. So that way you can predict in advance. Then there can still be some confounding factors, so or there can even be errors. So this goes on the negative side. So this is going to happen, but then you have no choice. So when you have some good benefit, you can consider some risk. And finally, there is improved therapeutic response, right? Because it is personalized medicine, and you know the patient has the required biomarker, and hence the patient is going to respond to the treatment. And especially if it is oncology, and if you are for sure that the patient is going to get benefited, then there is always an advantage, right? So these are some things I have got from the media. So this is about Washington DC, okay, and where there is a legislation getting introduced to prevent adverse drug effects, and this talks about personalized medicine. So something which the district or the state is going to do. Yeah, something very interesting. Gradually, many more countries and say even the states in the US all will come up with such things. Now I have one more video for you, and this is about accelerating precision medicine with artificial intelligence, and this is from Intel, and very interesting, slightly long video, but I'm sure you would love it. Let's get started, and I would make it full screen as well.
I really shouldn't be here today. I'm living a life that I didn't think I would get to live. My doctors told me that I had one of the most aggressive versions of prostate cancer that they'd ever seen. I didn't think I would see my daughter actually get out of elementary school. I went through surgery, chemotherapy, radiation. It didn't work. So now I'm willing to go outside the lines. Science and technology have come together to help us understand that there are unique drivers of disease in our DNA. Turns out that genomic sequencing saved my life. But my cancer started growing again after 2 years. And so now I need to figure out how can I stop it? And those answers aren't crystal clear right now. My name is Bryce Olson, and I'm the global strategist for Intel's Health and Life Sciences Group. So the standard of care for advanced cancer is really kind of a cut, burn, poison approach, but everybody's disease is really unique. So we shouldn't be treating every individual the same. I think it's tragic today that 96% of cancer patients just go onto the standard of care, and when that stops working, they die. When I got diagnosed with stage 4 prostate cancer, the cancer had spread into other parts of my body. That is when it becomes extremely lethal and dangerous. I knew that Intel had this group that was helping the healthcare industry use technology to make cool transformations. And so I learned about work that we were doing with genomic research centers like the Broad Institute of MIT, Harvard. Intel works with the Broad to really help them accelerate biomedical discoveries.
So the first human genome took about 10 years to complete, and now we're doing it in about 2 days. And that cost went from several $billion down to around $1,000.
The whole workflow is underpinned by Intel technology. Disease can really be diagnosed in the DNA. Genomic sequencing represented a molecular diagnosis. So I went to my pathologist and I had 2 words for him. I just said, sequence me. In our DNA are 6 billion letters. These are essentially the genetic code that make us who we are. Genomic sequencing allows us to uncover that genetic code and also to look for mistakes and defects in it that could be the drivers of disease. What we found is that I had mutations in my DNA that were messed up and causing my cancer to grow. I used that data to find a phase 1 early drug trial that offered the opportunity to shut down those specific drivers of my disease. And it worked. It shut it down for 2 years. That's what precision medicine is really all about. The right drug for the right person at the right time.
Oh! You're not done yet!
No, I'm not done yet. I love being a dad, and the only reason why I'm getting to live that life is because of precision medicine.
Did you?
But it's not so easy anymore. I started to build up some resistance to that drug, and my cancer started growing again. So we're now going to try to discover things that no one else has figured out yet, really. So I assembled a cross-functional dream team of researchers that want to partner with patients on these type of research projects to push the envelope, really, on what's driving somebody's disease and then try to help figure out an exploratory way to treat it.
So it's all automated. Wow.
The Broad Institute is one of the largest genome sequencing centers in the world. What we're doing is sequencing lots of people to try and understand the patterns of disease. Our genomic sequencers put out a huge amount of data. We manage about 70 petabytes of data. And to put that in perspective, if you look at every word in all of the books that have ever been written, it's still only about 50 petabytes. So we manage 20 petabytes more than that.
The problem is when you start amassing massive amounts of this data, there's things that we're gonna miss.
So Broad and Intel are working together to create AI and machine learning based tools to analyze genomic data.
When Intel thinks about artificial intelligence, we really think about how artificial intelligence can crunch that data to find a unique insight.
AI can find patterns in data that the human eye can't see, that machines can find.
As we get better at that into the future, AI can help unlock those insights and make it more streamlined for patients to get the right therapy they need as quickly as possible.
And that's gonna help someone like me fight cancer and actually beat it in the long run.
So I'm gonna have to hand you yours.
The interesting thing is that, if it wasn't for Intel, I wouldn't be here talking to you right now because they opened my eyes to this new way to fight cancer. When you get diagnosed with something like this, you can do one of 2 things. You can either go into a deep, dark hole, or you can try to take the limited time that you have and try to make it as meaningful as possible. Intel's technology is underpinning a whole revolution in making these new discoveries to really power a new era of healthcare intelligence. It's going to enable pharmaceutical companies to discover the next drug that potentially might even be in the timeline that I need. For me specifically, I've got a recurrence. It's pretty aggressive, but I'm not really that worried because I know how to fight cancer differently. Every advanced cancer patient who is running out of options should demand genomic sequencing because genomic sequencing can open up the door to a new way to fight cancer, and nobody should give up hope.
Now, I hope you found the previous video to be very interesting. Coming back to the original question that I asked you, that if each of us and our family members received the COVID-19 vaccine, based on our medical history, genetic profile, with guaranteed 90% plus efficacy and safety, right? So now after learning pharmacogenomics, you would agree with me that this is possible, right? Based on the genetic profile, you know which vaccine is going to work, right? So this is going to be the advantage, and this is the future of medicine. I trust you found this session to be useful and equally interesting. Feel free to reach out to us if you have any questions, comments, or any suggestions. All the best. Thank you.